This time I'm going to talk about membrane conductance. And by membrane conductance we mean the capacity of the membrane to conduct the transferring of ions from one side to the other. So by that we mean, for example, if we have a nerve cell, a neuron, and that neuron goes like that. Inside the neuron we have an electric potential, and outside the neuron we have an electric potential. This is extracellular and this is intracellular. And then we have the beginning of another neuron, which here let's assume that there are dendrites and this is the axon. So we have the axon, the cyton or the cell body, and then we have the beginning of the other neuron, and this region around here is called the synapse. Now this membrane conductance, which involves the exchange of ions or the movement of the ions from one side to the other, of course would be done through some certain specific structures that would allow this process. And these structures are called channels. So these structures are called channels. And we have three types of channels. The first type of channels are called, so let's say, okay, the first one, let's assume that this is a channel, it's called ungated. So ungated means that it's an open channel. There's no gate on this channel. An example for such kind of channels are potassium leakage channel. So from the name it's called the leakage channel, it's always leaking potassium. And we have to know that the concentration of potassium is around 140 millimoles. So inside, so intracellular potassium is around 140 millimoles. Outside, potassium is around 5 or 4 millimoles. So this kind of channels is called ungated because there's no gate on it, it's always leaking. And this kind of channels is formed, or it's found in all types of cells, especially for potassium. And then we have another type of channels. We have voltage gated channels. So voltage gated channels. So what does this mean? It depends on the voltage. Now, in the resting state, the electric potential inside the neuron will be around, so resting, it will be around minus 70 millivolts. When a stimulus comes, this resting potential will jump to around positive 30 millivolts. This change in the voltage will cause the opening of these channels. So they are gated by the voltage, the change in the voltage. An example for these channels are sodium channels. Sodium channels. So whenever this happens, sodium starts to go inside the cell. We have to know that sodium in the extracellular fluid has a concentration of about 142 or 100 millimoles. These values are different from one book to another, and inside it's around 12 millimoles. So this is the second type of channels. The last type of channels, let's assume when the stimulus reaches the end of the neuron and it wants to cross and pass to the next neuron in order to stimulate it. So here we have sacs, which when the stimulus arrives, calcium will go inside the cell, these sacs will rupture, and they will release something called a neurotransmitter. For example, a very important neurotransmitter is called acetylcholine. For short, ACH. This acetylcholine will bind to our third type of channels, which are called ligand-gated or ligand-gated channels. Ligand-gated channels. So these ligand-gated channels will be activated by the binding of acetylcholine. So when acetylcholine binds to this channel, it will open, allowing sodium to go inside the cell, which will lead to the propagation of the stimulus, which was originally in the previous neuron. So now we know our three types of channels. Now, the last point that I'm going to talk about is the net force. The net force of this membrane conductance. What does it depend on? It depends on two independent factors. So both of these factors are not depending on each other. The first factor, which is the concentration force. So the concentration force means that we have a concentration of the ions inside and we have the concentration of ions outside. Whenever the concentration is higher, the ions will move from the higher concentration to the lower concentration. So this is the concentration force. The second concept, or the second factor, is the electrical force. And the concept here is very simple. We have a number and we have a sign. So the number estimates the value of the actual voltage, and the sign would let us assume the nature of the electrical force. So let's say if it's a negative 70 millivolts, so it's a voltage of the power of 70 millivolts with a negative charge, assuming that it will attract all the positive charges and it will repel all the negative charges. So this voltage will prefer attracting positive charges, or in other words positive ions, like let's say potassium, sodium and so on, and it will repel negative ions like chlorine. And these are the two factors that affect the force of the net force of the membrane conductance. So this is it for today. Next time we're going to talk about a concept which is called the Nernst potential that would explain why we have a resting potential. So we're going to talk about this in the next video. Until then, I thank you for watching and see you.